Bismillahirrahmanirrahim. Assalamu alaikum dear friends. Today we will discuss about the erythropoiesis. It means formation of the RBCs. In previous classes we have discussed about the blood, component of blood, RBCs, WBCs and platelets. But in this class we will discuss about the only formation of RBCs. It means erythropoiesis. If you are watching first time my YouTube channel, kindly subscribe and press on the bell icon for the new updates. So let's start. In bone marrow, so many pluripotent cells are present. If we take a single cell, after that single cell will divide it into two cells, lymphoid stem cell and a myeloid stem cells. And myeloid stem cells further divided into two cells and they are colony forming unit, granulocyte monocytes and colony forming unit erythrocyte and megakaryoid. And after that this megakaryoid cell divided into two cells and they are megakaryopoiesis and colony forming unit erythropoietin. And this megakaryopoiesis will form the platelets and a colony forming unit erythropoietic cells will further convert into proerythroblast, and this proerythroblast is very large in size and it has much large nucleus, open nucleus with loose chromatin, and the cytoplasm is bluish. After that, this proerythroblast will convert into basophilic erythroblast. Why we call it basophilic? Because the color of cytoplasm is much blue than the proerythroblast and the nucleus is short than the proerythroblast. And the formation of ribosomes occur in this basophilic stage more than proerythroblast. After that, this will convert into polychromatic erythroblast. Poly means many and chromatic means colors. That means in this stage, the cell produce in much amount globulin protein and the chromatins which are present in the nucleus they start condense. That means they will condense and become heterochromatic, and then this cell will convert into orthochromatic normocyte, and after that this cell will convert into reticulocyte. It means after the removing of nucleus some network like blue network appear or retics appear in the cell. That's why we call it reticulocyte, and they remain 1 or 2 days in the circulation. After that they will leave the cell and the cell become mature RBC, and it will running in the circulation up to 90 to 120 days. Erythropoiesis. That what is erythropoiesis? Erythropoiesis is a process by which red blood cells are formed in the bone marrow. This process starts from the pluripotent stem cells which are present in the bone marrow. Single pluripotent stem cell will divide it into two cells, lymphoid stem cell and a myeloid stem cells. The myeloid stem cell further categorized into colony forming unit granulocyte monocytes and colony forming unit erythrocytes megakaryoid. And a megakaryoid will convert into megakaryopoiesis forming it means that megakaryopoiesis form the platelets and a colony forming unit erythropoietic cells, and it will change into erythroblast. It will means the colony forming unit erythropoietic cells. They will change into proerythroblast. This is proerythroblast. In proerythroblast, nucleus is very large than with very fine chromatin. That means the nucleus of this proerythroblast is very large and with a very fine chromatin. Very fine chromatin means loose chromatin. Here loose chromatins are present. It means not condensed or not heterochromatins are present here. And a prominent nucleoli which form the ribosomes. And the nucleoli are present in the nucleus of proerythroblast, which form the ribosomes, and ribosomes synthesize the proteins, mainly hemoglobin proteins, and they come into the cytoplasm. It means these nucleoli produce the ribosomes, and ribosomes come into the cytoplasm for the synthesis of the proteins, mainly hemoglobin. It has open nucleus. Open nucleus means nucleus with loose chromatin called euchromatin. These are the loose chromatin. It means they are separate from each other. So we call it euchromatin also. Cytoplasm is basophilic. It means normal blue. The color of cytoplasm here is normal blue due to the less concentration of the ribosomes. The blue color of cytoplasm is due to the ribosomes and mRNA. That means the basophilic color produced by the ribosomes and mRNA. As the concentration of ribosome increased in the cytoplasm, it becomes more blue called basophilic erythroblast. That means when the high concentration of ribosomes are produced in the cytoplasm, then the color of cytoplasm become much blue, more blue than this color. So at that stage is known as basophilic erythroblast. Now come to the basophilic erythroblast. In this stage, you will find that some heterochromatins are appeared, and the color of cytoplasm is much blue due to the high concentration of the ribosomes, and the size of nucleus is also reduced, and the whole cell also reduced than the proerythroblast. As the concentration of ribosomes increase in the cytoplasm, it becomes more blue called basophilic erythroblast. That means when concentration of ribosomes increased in the cytoplasm, then the color of cytoplasm change into much blue, and that stage is known as the basophilic erythroblast. Open nucleus becomes short. It means the nucleus start going to shorten, and the chromatins also condense. The new open nucleus becomes short, contains loose chromatin and some heterochromatin. It means some heterochromatins are formed or condensed chromatin. Due to the high concentration of ribosomes, some red proteins, it means hemoglobins, appear in the cytoplasm. It means when the concentration of ribosomes increase in the cytoplasm, then some red proteins, it means hemoglobin produced in the cytoplasm, appear in the cytoplasm. Then the stage is known as a polychromatic erythroblast. When some red proteins produce in the cytoplasm, then that stage is known as polychromatic erythroblast. So let talk to me about the polychromatic erythroblast. In this stage, you will find that some red proteins are produced here, and the chromatin condensed here. It means heterochromatins are formed here. The nuclear size is much short than the basophilic erythroblast and proerythroblast. Poly means many, chroma means colors. It means many colors produced in this stage. In polychromatic erythroblast, blue and red colors appear. Blue color show the ribosomes, and red color show the hemoglobins. Blue color shows the high concentration of ribosomes and red color shows the hemoglobin. And this stage, nucleus is very short and contains totally heterochromatin. Here no loose chromatins are appear or remaining, but whole chromatins are condensed and become heterochromatin. Now cytoplasm is going to hemoglobinized. That means much concentration of ribosomes produce much concentration of the hemoglobin. It means proteins. So now the cytoplasm will become much hemoglobinized. Then that stage is known as orthochromatic normocyte. In orthochromatic normocyte, the nucleus is very short and condensed chromatin. After that, this nucleus will remove the cell. Orthochromatic normocyte is much mature because so much hemoglobin are formed. That means in the orthochromatic normocyte, much hemoglobins are produced and the nucleus become very short. Nucleus is very much short and contain totally heterochromatins. After that, nucleus leave the cell and become totally hemoglobinized. When nucleus removed from the cell, then that cell is totally hemoglobinized. It means in that cell totally hemoglobin is present, not nucleus. After some time, the biliretics appear like network so-called reticulocyte. After some time, the blue dotted structures, it means the network-like structures are produced in the orthochromatic normocyte. Then that dotted lines are the retics, and that's why we call it reticulocyte. And reticulocyte remain 1 or 2 days in the circulation. After that disappear, and the cell become mature RBC. Look in this diagram. This is a cell. It means orthochromatic normocyte. In this stage, the cell become totally hemoglobinized and contain very condensed mature nucleus. And now this nucleus is ready for the living cell. After that, this nucleus will remove from the orthochromatic normocyte, and this cell will convert into the this structure. And after some time, blue retics are appear in the center of cell, and these blue retics called reticulocyte. When blue retics appear in the cell, then that stage or cell is known as reticulocyte due to the presence of retics. And then these retics will remain 1 or 2 days in the circulation and leave the cell or disappear from the cell, and cell become totally hemoglobinized or become much mature RBCs. So dear friend, this was about the formation of RBCs or erythropoiesis. Khuda Hafiz, wassalam.